Chapter 4, where we will discuss the history of life on Earth as well as the genetics of evolutionary processes. In the previous lecture, we examined how species form. We know that these speciation events can occur rapidly or they can occur gradually over time. We also know that small genetic differences can create large differences between organisms and lead to the formation of new species. We are now going to take a look at the genetic basis for evolution. Many species have evolved due to small changes in developmental genes. These small changes can cause large-scale changes in body form. This is because many of these developmental genes are involved in regulating either the rate, timing, or spatial organization of an organism during development. Heterochrony refers to a change in the timing or rate of developmental events. This can cause major changes in the adult form and can even lead to paedomorphosis. This occurs when an adult is reproductively capable even though it has juvenile or ancestral features. Heterochrony in the time taken to development can drastically change the formation of major body structures. An example of this is looking at the development of the vertebrae in giraffes. All mammals typically have 7 cervical vertebrae, except for manatees and sloths. However, because giraffes experience an extensive growth period and prolonged gene expression of these developmental genes, this has allowed for the elongation of the cervical vertebrae in giraffes. Similarly, small changes in gene sequences can also cause large-scale changes in body form. This is because changes occur in every single cell that expresses that specific gene. An example of this involves Hox genes or homeotic genes. These genes are considered master regulatory genes that are commonly involved in determining the spatial arrangement of an organism. Here in the diagram below, we can see how a small change in the gene sequence completely changes the functionality of that gene. Hox gene 7 in crustacean ancestors, when expressed, does not suppress legs. As a result, we see the formation of legs on many of the body segments. However, due to changes over time within the gene sequence, now in the present-day insects, the expression of the Hox gene causes the suppression of legs. This is commonly seen in the development of the abdominal region, where we do not have the presence of legs. Legs are restricted to the thorax region. The following example demonstrates how small differences within genomes can cause a large amount of diversity. If we examine the human and chimpanzee genome, we note that they differ only by 1.2% of their base pairs, and 2.7% are due to insertions and deletions. Many of these differences are due to duplications of genetic sequences, rearrangements of sequences within a chromosome or between chromosomes, as well as mutations. The diagram to the right gives you an overview of what the human genome consists of. Note that only 1.5% of our genome is dedicated to exons or expressed information. The other 5% consists of regulatory sequences, as well as introns, unique non-coding DNA, as well as repetitive DNA sequences. These repetitive DNA sequences comprise the majority of the genome. These include transposable elements, related sequences, and unrelated sequences. Gene fusion can also result in the formation of new chromosomes. If we take a look at chromosome 12 and 13 in chimpanzees, it bears a distinct resemblance to chromosome 2 in humans, even including the telomere-like sequences that are located at the centromere as well as at the tips of the chromosome. In addition, chromosome 16 in humans contains various regions that are similar to chromosome 7, 8, 16, and 17 in mice. This suggests a divergence from a common ancestor. Transposable elements are stretches of DNA that can move to different regions within the eukaryotic genome. These include transposons and retrotransposons. Transposons move via DNA-based intermediate. This typically results in an original transposon as well as its copy. Retrotransposons, on the other hand, move via an RNA intermediate. This means that they need reverse transcriptase to create a DNA copy that is then inserted into the genome. Unequal crossing may occur during meiosis or slippage during DNA replication, which can lead to deletions and duplications within the genome. We've typically seen that during meiosis, homologous chromosomes pair up to form a tetrad. However, incorrect pairing of two homologous chromosomes may occur. This may cause crossing over of misaligned non-sister chromatids. Many times, these transposable elements can serve as crossover sites between these non-sister chromatids, allowing for easier exchange of genetic information between non-homologous chromosomes. It is also noted that the duplication of ancestral genes provides a template for multiple mutations and divergence. An example is looking at the human globin genes. Many of these genes derive from one ancestral globin gene that had duplicated and diverged approximately 450 million years ago. This has now resulted in many gene sequences in different locations within the genome with diverse but related functions. The diagram to the right demonstrates the duplication of an ancestral globin gene. This then led to the alpha and beta globin gene families on different chromosomes. Many of these genes are involved in binding or transporting oxygen within organisms. In some instances, a mutation can lead to a novel function for a gene. An example of this involves the FOXP2 gene. This is a common ancestral gene that is known to regulate genes for proper brain development as well as being responsible for vocalization in mammals. In a study examining the function of the FOXP2 gene, as we can see in the image to the right, we can compare the functionality of this gene by examining wild-type, heterozygotes, and homozygotes. Wild-type has both normal copies for the FOXP2 gene. A heterozygote has one disrupted copy, and a homozygous has two disrupted copies of the FOXP2 gene. We can note in the chart below that the vocalization sounds within mice was severely disrupted anytime that the FOXP2 gene was mutated. Once again, furthering the importance of the FOXP2 gene in vocalization. What is noted with the FOXP2 gene is that there are distinct differences between the human and chimp genes. This suggests that a mutation may have arose that led to the evolution of speech in humans. It is also noted that identical sequences in humans and Neanderthals have been found with the FOXP2 gene. This suggests that Neanderthals may have been capable of speech. Exon duplications and shuffling due to meiotic errors can also lead to the formation of new proteins. This is demonstrated by examining the TPA, or tissue plasminogen activator gene. The TPA gene commonly produces a protein that assists in the process of blood clotting. The TPA gene may actually have originated from exon shuffling from 3 different genes. There are genetic similarities in the TPA gene to epidermal growth factor gene, the fibronectin gene, as well as the plasminogen gene. Due to exon shuffling, the TPA gene exists as it does today. An important trend to note is that highly important regulatory genes tend to be conserved both throughout evolution as well as between species. This highly suggests that these genes are essential for life. An example of this is once again looking at the HOX genes, or homeobox-containing genes. Remember, these genes are highly important in regulating the overall body organization of many different organisms. Many of these HOX genes themselves contain similar DNA sequences. We can refer to these sequences as homeobox regions. These regions are highly conserved. In fact, they are identical or very similar both within vertebrates as well as invertebrates. And many of these related sequences can also be identified in yeast, plants, as well as prokaryotes. We can take a look at the diagram to the right to see the presence of Hox genes in both the human as well as the Drosophila. Homeobox genes also demonstrate the concept of collinearity. This occurs when the spatial organization of these genes reflects both their timing of activation as well as their expression. Early genes are commonly located at the anterior part of the organism, whereas later genes are typically located posteriorly. We can see the distribution of various types of Hox genes within the fruit fly as well as the mouse embryo, also demonstrating a pattern of organization that is also highly conserved, where the purple genes are expressed anteriorly and the red or orange genes are expressed posteriorly. In addition, small changes in either the timing or pattern of gene expression can cause drastic changes in body form. Once again, we examine the expression of Hox genes. We consider 4 different Hox genes expressed in 2 different organisms, each depicted by a different color: purple, yellow, orange, and teal. We can see in the shrimp Artemia we have the expression of 3 Hox genes that are expressed simultaneously. This leads to the formation of many leg-bearing appendages along the body, as well as the expression of the 4th gene in teal, which leads to the formation of the genital appendages. We can see a very distinct pattern of expression in the grasshopper. In the grasshopper, each of these Hox genes is expressed discretely in specific areas and leads to the formation of very distinct features, such as the fore and mid legs, the hind legs, the abdomen, as well as the genital features. It is important to remember that evolution is not considered directional. That means that evolution does not simply move towards an organism becoming or a feature becoming more complex. It doesn't strictly dictate that a species has to change. Rather, it simply reflects an organism's ability to be best suited or best fit for its environment. For instance, many creationists argue that the horseshoe crab is considered to be a living fossil, and that its design and lack of change over time since the fossil record strictly conflicts modern-day evolutionary theory. However, if you take a closer look, you will note that there are some morphological adaptations that have occurred, as well as various speciation events that have occurred within this group of organisms, thus demonstrating that although there aren't many changes, evolution is still occurring. Another example of this concept is looking at the evolution of the eye. Many creationists argue that the eye is increased in complexity to form the eventual human eye, which is considered to be the most complex form. However, this is not always the case. In some instances, a simple eye can be more useful to an organism than a complex eye, and vice versa. We see that in limpets, a conglomeration of photoreceptor cells work just as well for them in their environment as the complex eye does in the squid. In addition, we can see that many different types of complex eye exist that are better suited for an organism in their type of environment, thus re-illustrating the fact that evolution is not working towards an increase in complexity in structures, rather an increase in the suitability of structures for an organism to function the best in their environment.